Hello friends, today I'm here to discuss regarding a summarized video on body fluids and circulation. Blood components. The blood components majorly consist of red blood cells, platelets and WBC and plasma, out of which the plasma comprises of 55%, RBCs are 44%, and platelets and WBCs account for 1%. The plasma is divided into for 91% of water, 7 to 8% of proteins, that is albumin, globulin, and other components up to 2%. The blood groups. There is a B, O blood types, blood group discovered by Karl Landsteiner in 1901, which is universally followed. This ABO blood group is a description of a genetic locus that has three alleles, that is IA, IB, and IO, which determine the blood group, where I stands for immunoglobulin. The type O allele is a functionless and recessive, while type A and B alleles do cause strong agglutinations, and these are the decisive alleles which have a decision making in whether the blood group is A, B, AB, or O. These are the four categories: Group A, B, Group AB, and Group O, which are recognized and followed globally. Red blood cell types which have Group A, which are called to be Group A, they are having antibodies of B, anti-B present, and A antigens present on them. Group B has antibodies for A present on them and a B antigen, while AB have no antibodies on them and have A as well as B antigens, while O, they have antibodies for both A and B, while there are no antigens present on them. But how this blood transfusion exactly takes place? I'll be showing you a small animation video, which is obtained from a outsource. This A plus body, where the A plus can be delivered to A as well as AB positive. A minus are given to A plus, A minus, AB plus, and AB minus, while B plus can be transfused to B plus and AB positive people. B minus, B negative, it can be given to all B plus, B minus, AB plus, and AB minus. O positive blood can be transfused to all positive blood groups, O, A, AB, and B, while O negative can be given to all the blood groups, that is O plus, minus, everyone. AB plus can be given only to AB plus blood group people, while AB minus can be given to both AB plus and AB minus. With this short video, we can conclude that the blood group AB plus is the universal recipient, while the blood group O negative is the universal donor. They can give blood to everyone in the world, O negative, while AB plus, they can take blood from everyone which are coming, and AB minus too. Now the blood coagulation, the clotting factors. This is a clotting factor mnemonic, which say that the blood group 1, 2, 3, 4, which are 13 in number, have given as foolish people try climbing long slopes after Christmas, some people have fallen. With this mnemonics, we can try to remember that F stands for fibrinogen, P stands for prothrombin, T stands for thromboplastin, C for calcium, L for labile factor. 6 is no longer used in the scientific studies. S is for stable factor, A, antihemophilic factor A, C is the Christmas factor, S is for Stewart power factor, P is for plasma thromboplastin, HFC, H is a Hageman factor, and F is fibrin stabilizing factor. All these 13 factors are responsible step by step on the clotting mechanism. But how exactly this clotting mechanism takes place? It is a complex process of enzymatic reactions, and these enzymes and the clotting factors which are activated together, they are sequentially resulting into the clot formation. There is 3 stages of coagulation, that is intrinsic or extrinsic, thrombin formation, and fibrin formation. With the short video, we'll be seeing that how blood actually clot takes place. When any injury takes place to a site, at the site of cut or any injury, there is a bleeding. This bleeding is actually resulting into the cut in a small artery, and from this small artery there is a red blood cells which are carrying the oxygen. There you can find platelets, which are flowing in the blood. We can find the WBCs and the clotting factors, which are actually moving around in the blood. And this damaged blood loss leads to oozing out of blood. Now, how this blood clot will form and the blood injury will be sealed. This clotting mechanism is actually takes place when there will be adhering of these platelets towards the site of injury, and the cutting edges, there is a platelet plug forms, and which causes the external bleeding to stop. And the clotting factor, they create a cascade of reactions, because of which a fibrin is formed, and this fibrin stick together and seal the wound. And then day by day, sequentially, and eventually the blood clot makes it normal as it was earlier. So this is how a blood actually forms. So we'll see what are the further details coming in this process of blood formation. This is a diagrammatic view of how the injury takes place and the blood oozes out from the broken vessel of the blood, which is taking place at the site of injury. The platelets are the active role playing material over here. They activate the mechanism, and when the clot is formed and the fibrins are formed, they actually seal the site of injury, due to which the blood flow stops. This coagulation mechanism is given diagrammatically by these pathways. When there is a danger signal which is given over here is a site of injury, the platelets, they get aggregated. The tissue factor 7, it activates 7A. 7A, it causes conversion of 10 factor to get activated in presence of factor 8, which is then leading to activation of factor 5. Factor 5A together converts the prothrombin to thrombin, and this thrombin converts fibrinogen to fibrin, and here there is a clot formation takes place. This is a mechanism of how step-by-step blood coagulation take place. And a short video is given here, which is an animated video which will show how this cascade actually works, so that it will be easily understandable by my viewers and all of us who are studying coagulation. This white cells which are seen here are the blood platelets. They are getting activated, and this platelet coagulation causes accumulation of different factors which come together and form a fibrin. We'll see step by step how this activation takes place and how the clot is formed. The tissue factors, they form complex with 7, which is an activated 7A. The 7A leads to activation of factor 10 to form 10A, which is an activated form. And this activated factor 10, it leads to extrinsic pathway. In intrinsic pathway, there are 12A, 11A, 9A, 8A. A chain reaction takes place in intrinsic pathway to form a complex with 10, which is a common factor now, intrinsic and extrinsic. This 10A, which is coming from extrinsic as well as intrinsic factor A, which will cause an important role in coagulation. This 10A activated, and 5A will cause prothrombin to get converted into thrombin. Now the thrombin is getting activated by this complex of activated 10 and 5 factor, and this thrombins together will form so many molecules of thrombin in the blood circulation, and which will get released, which will cause more platelets to get activated and aggregation, and they will form fibrin to get formed. This thrombins will form strands of fibrin which cause fibrin clot and stabilize the clot so that the injury gets sealed enough. So this is a complete pathway where the blood cells are getting held together to form a platelet, and the 10A is a central role which is common to both extrinsic as well as intrinsic pathways, and this 10A leads to thrombin activation and later on fibrin formation. This video is taken from the Bears. The credit of this animation goes to them, which have helped us to understand it so easily, the coagulation cascade. Now moving towards the next body fluid apart from blood, that is lymph. The lymph is a colorless fluid component of the lymphatic system, which flows through the lymph vessels. The lymphatic system's function is to return the fluid from tissues through to central circulation through the nodes. Lymphatic system consists of three major components, that is lymphatic vessels, spleen and tonsils. These three major components are there which govern the functioning of the lymphatic system. The lymph capillaries in the tissue spaces, they are activated and they flow in the spaces between the cells of the body tissues, which are small just like holes in a sponge. And this fluid is then transferred to lymph capillaries. Lymph consists of cellular as well as non-cellular part. The cellular part consists of mostly lymphocytes and other WBCs, but mostly lymphocytes are active here. And the non-cellular part consists of majorly 94% of about 94% water, and other 6% includes proteins, fats, carbohydrates, enzymes, and antibodies. The functions of lymph includes the supply nutrients and oxygen to the places where blood cannot reach. They drain the excess fluid from tissue spaces and send it back to the blood. They help in absorption of fats and by lymphatic vessels in small intestine. Then they contain monocytes and lymphocytes, as we have seen earlier, majorly lymphocytes and monocytes from the other WBCs. They help in defense and they eliminate bacteria and other pathogens from the tissues. Human circulatory system. Next to the lymph, we'll shift to human circulatory system, which is centrally controlled by the heart as the pumping organ. What happens in the heart is the superior and inferior vena cava, they act as an inlet to the deoxygenated blood which comes inside to the heart in the atrium, right atrium, and then from the right atrium it goes to the pulmonary artery, from where it is moved towards the lungs for getting oxygenated in the I'm sorry, in the lungs. The pulmonary system of the lungs, which takes oxygen from outside and liberates the carbon dioxide which is coming from the blood, and they oxidize the blood which is coming, and the oxygenated blood enters through pulmonary vein into the left atrium. From the left atrium in the left ventricles, and from the left ventricles it moves towards the aorta and then circulated through different cells and tissues of the body. This is the anatomical structure of the heart, in which we will be studying the conducting system too. Here you can see what I was describing in the previous diagram, the superior and the inferior vena cavas, which will be taking the deoxygenated blood. This is the bicuspid valve, and this is the tricuspid valve, which act for the opening and closure gateway of the blood which is entering through the vena cavas, the deoxygenated blood in the right ventricle and the oxygenated blood in the left ventricle. From the left ventricle, when the blood is shifted to the semilunar vesicles, they will be semilunar valves, sorry, they will be shifted to the either in the pulmonary vein to get pulmonary artery to get oxygenated, and through the aorta, the oxygenated blood will be circulated to different parts of the body. Now, how this conduction actually takes place? There is a pacemaker located in the right atrium in the heart, the SA node, which generates the conducting system and transfers the conduction from SA node to the AV node at the junction of the atria right and ventricle right. And from this, the conduction is passed to the bundle of His. From the bundle of His, which is located in the septum of heart, central septum of the heart, and from this it is transferred to the Purkinje fibers, and then a systolic pressure is generated which contracts the ventricles, and the blood is transferred to the aorta, to different parts of the body, and through the semilunar valve it enters into the pulmonary artery, and the deoxygenated blood is sent to the lungs for getting oxygenated. So this is how the conduction actually takes place. But this conduction and control of heart together is governed by a cycle, the term which is used to describe the relaxation and contraction which occurs at the heart and which works to pump the blood through the body. And the cardiac output is defined as the volume of blood actually pumped by the heart each minute. It is circulated as a heart rate into stroke volume, that is the time the heart is pumping into the volume of blood which is oozing out in each minute. This is the cardiac output. So this is the ECG diagram, a normal ECG which can be studied from here. It is a P wave, a baseline comes and a QRST complex. And this ST complex is, the P is the initiation of the, it is coming from the atria. It is the initiation of the SA node. Then at the AV node, when AV node and the Purkinje fibers and bundle of his are actually together working for contraction, where that is the systole. It is coming from QRS complex, and then the T wave, which is coming, which is a diastolic movement where the plateau region is seen and a relaxation can be seen. We'll be studying from this link of Wikimedia, which will be showing us how ECG actually work as a principle. This is a small GIF which is available from the Wikimedia for a complete study. We can see from this diagrammatic view that how after this AV, the SA and AV node, they actually work, and how the ECG pattern is generated. Like see how the SA node first gets activated. Will the SA node, when on activation, it will cause the P wave to generate. SA node causes the atria to get contracted and the P wave is generating. Then AV node takes the charge, and AV to Purkinje to bundle of his and Purkinje fibers, the QRS complexes form, and the Purkinje fibers, it forms a diastolic form comes over here, in which relaxation takes place. So how this conduction and ECG can be understood together by this small animated GIF, which is available with us from Wikimedia. We are thankful for the source to them, as they have helped us in understanding this media of ECG, which is coming. Then studying regarding the ECG, we have understood how exactly it takes place, and this is the study in which we can see the normal functioning of heart. If any change in the P wave or QRS complex, or a change in the duration of this, which is taking a duration of 0.8 second, this cycle is a mere of 0.8 seconds. So we can understand the disturbances where it is actually taking place by this ECG, whether the atria is affected, whether the ventricles or the segment of heart muscles are affected, or the diastolic relaxation is affected. So we can study with this and understand the ECG. So this is all together what we have studied regarding the lymph, regarding the body fluid as a lymph, and we have studied the blood, its components, and the different blood groups. It is a brief description for all of us over here. And with this, I take a bit from the audience over here. Thank you very much, so that we can continue our study in coming days too. Hope to see you soon.